Hello friends, welcome in online learning session. Myself J.Y.Mote, working as Assistant Professor, Department of Animal Husbandry and Dairy Science, College of Agriculture, Nashik. Today we are going to study about system of breeding under the course AHDS 242. First, we will see what is meant by breeding. The term breeding defined as the manipulation of genetic material in accordance of requirements of the terms dictated by the demand, called as breeding. This system of breeding have been broadly divided as under two classes. First one is inbreeding, second one is outbreeding. Inbreeding means mating of closely related individuals or animals, whereas outbreeding means mating of unrelated individuals or animals. The system of breeding mainly classified into two classes, that is inbreeding and outbreeding. Then inbreeding also divided into two classes, close breeding and line breeding. Then outbreeding also divided into four classes, that is outcrossing, crossbreeding, breeding up and species hybridization. Then crossbreeding divided into four classes, that is rotational crossing, back crossing, two breed crossing, three breed crossing. Then rotational crossing also divided into two classes, that is Criss Crossing and triple crossing. We will see first one by one. First one is inbreeding. Inbreeding is the mating of related individuals within 4 to 6 generations. In other words, we can also define as mating of more closely or distantly related individuals than the average of the population. It means that more closely related animals are mating here, called as inbreeding. Close related individuals such as selfing, full brother, sister mating, etc. Mother to son mating, father to daughter mating, while distantly related individuals, that is cousin mating, cousin brother, cousin parental uncle, or niece mating, parental aunt and nephew mating. Advantages of inbreeding: increases in homozygosity and decreases in the heterozygosity. It helps in the producing seed stock to be used for crossbreeding. Then it helps in the development of families and inbreed lines. It is the only known method of increasing prepotency. Prepotency is the ability to transmit characteristics to the offspring to a marked degree. Also, it helps in the elimination of undesirable recessive gene or characteristic in the individuals. It helps to obtain purebred animals which are genetically uniform, which are crossed. It helps in the selection of desirable and culling of undesirable animals. It increases uniformity in productive characteristics or characters. Then disadvantages: it decreases growth rate, body size and body weight. It reduces reproductive efficiency and general vigor because of mating of closely related animals or individuals. It delays puberty in both the sexes, that is male and female also. Then it increases early embryonic death rate. It reduces adaptive traits to adverse condition and fitness. It increases mortality in the progeny. It causes drop on milk production in cows, wool production in sheep, and eggs production in poultry. Also, it increases susceptibility to infection and adverse environmental factors. Inbreeding can be again divided into following groups, that is close breeding and line breeding. First one, close breeding. In, in this, mating of full brother and full sister or sire and daughter, whereas line breeding, half brother and half sister, means closely related animals. First one is close breeding. Close breeding means the mating of very closely related animals than the average in the population. For example, the mating of full brother and full sister, sire to his daughter, dam to her son, etc. This type of mating should be used only when both parents are outstanding individuals. Then advantages: it helps in the detection and elimination of the harmful or undesirable recessive genes. It develops uniform progeny. Also, it always used for testing of breeding pool to declare him free from undesirable or deleterious recessive genes and brand him as proven bull. Second one is line breeding. The mating of animals wider degree of relationship than those selected for close breeding, called as line breeding. It is a mating of two pure lines with a specific purpose. Line breeding means mating of distantly related animals. It involves mating of grandson and granddaughter or cousin mating. In advantages, increase in uniformity of characters. It slowly increase homozygosity and hence less risky. It slowly develops undesirable characters if present in the population. The disadvantages. It gives less benefit for few generations because animals are selected on the basis of pedigree record, giving no importance to real individuals' merit. The next one is outbreeding. It is a system of mating of individuals who have no ancestor in common in the first 4 to 6 generations in their pedigree. Hence, distantly related animals are mating here. In other words, in the mating of unrelated individuals within a breed. This kind of breeding system brings fast improvement in the herd or breed. Faster growth rate achieved throughout the breeding. Then types of outbreeding. Outcrossing, crossbreeding are the main types of outbreeding. Then crossbreeding also divided into rotational crossing, triple crossing, back crossing. Then species hybridization, breeding up. First one is outcrossing. Outcrossing means mating of unrelated but pure breed animals within the same breed. That is gir cow or gir bull, which are unrelated. It involves mating of animals which do not have common ancestor in the first 4 to 6 generations in their pedigree, and the offspring of such mating is known as the outcrossing. Advantages. This method is highly effective for characters that are largely under the control of with additive effects. Examples are milk production, growth rate, staple length, and fleece. It is an effective method for genetic improvement of the cattle within the breed. Then crossbreeding. Crossbreeding is the mating of animals of different breeds. Example are Holstein Friesian sire mated with the or crossed with the Geritan. Here two breeds are crossed. Crossbreeding produces hybrid figure or heterosis, a combined effects of epistasis, dominance, and additive gene action, etc. cause hybrid figure. Crossbreeds are mainly used for production and improvement in the efficiency of production traits. It is generally used for production of new breeds. Then heterosis or hybrid figure. It is the phenomenon in which the crossing of unrelated individuals results in the progeny which is superior to the parents, called as heterosis or hybrid figure. Method of crossbreeding. Rotational crossing. First, Criss Crossing. When two breeds are crossed alternatively, the method is known as Criss Crossing. This method is proposed for utilizing heterosis is both dams and sire. Here, two breeds are crossed alternatively. That is, A crossed with the B, then F1 is also again crossed with the A, then F2 crossed with the B. Likewise, then triple crossing. In this system, three breeds are crossed in the rotational manner. In this system, three breeds are crossing like A crossed with the B, then F1 is crossed with the C, then ABC is a 25-25 to 30 ratio, again crossed with the A. Likewise, then back crossing. Back crossing is the mating of cross animals back to one of the pure parent races which were used to produce it. It is commonly used in the genetic studies, not widely used by the breeders. Here, tall and dark, two breeds are crossed with each other, where F1 is again crossed with one of its pure parents. The phenotypic ratio of all tall to dwarf ratio is 1:1. All being tall, there is no ratio of tall to dwarf. Two breed crossing method is mating of two different breeds, two crossbreeds. That is Holstein Friesian crossed with the Devni. The F1 is produced whole Dev. Then three breed crossing. Here Fule Triveni is a triple breed cross or three breed cross, where Jersey crossed with the Gir having 100% as to 100%. Then F1 Gir Jersey having composition of Jersey and Gir, it is 50:50, crossed with the HF, that is Holstein Friesian. Then F2 is produced Fule Triveni having 25:25 to 50 ratio. And advantages of crossbreeding, that is, it is available as a means of introducing desirable characteristics into a breed in which they have not existed formerly. It is useful in producing new breeds. It increases the hybrid figure, growth rate, fertility, and production in the progeny. It helps to reduce animal with the better adaptability in adverse environmental conditions with the increased resistance to diseases. It is an extremely handy tool to study the behavior of the correct characteristic in hereditary transmission. The disadvantage, it requires maintenance of two or more pure breeds. It may sometimes produce progeny with undesirable traits also. It increases heterozygosity and thereby reduces breeding merits of the progeny. Next one is grading up. Grading up is defined as mating of poor breed sire with non-descript females and also with offspring for generation after generation till the progeny reach nearly 98 to 99% of the original breed, called as grading up. The continued use of good pure breed sire for only a few generations are all that require the herd to the point at which it has all appearance, actions, and practical values of the breed. The advantages of the grading up. It is the best method of improving the local non-descript animals within the reasonable time and expenditure. It helps develop pure breed after few generations. It is simple and economic methods of improving herd of the pure breeds. Then it helps to prove the quantity of sire and thereby increase its market value. Then disadvantages. Poor breed are not always better than the indigenous animals. Then pure breed from temperate zone may not give good result in the tropical climate. Male offspring are not suitable for breeding purpose. Then species hybridization. It means mating of animals of two different species called as species hybridization. It is extreme from outcrossing. Example are Jack, that is male ass, crossed with the mare, that is female horse. F1 is produced mole. Then stallion, that is male horse, crossed with the jenny, that is female ass. F1 is produced hinny. Whereas female zebra crossed with the stallion, F1 is produced zebroid. Advantages of species hybridization, that is, it produces progeny having more vigor, disease resistant, and hardy than the parental species. It produces progeny which shows better adaptability in the adverse environmental conditions. Disadvantage, progeny is usually sterile. The difference between inbreeding and outbreeding. Inbreeding means mating of related animals, whereas mating of unrelated animals in outbreeding. Example are mating of dam to son and sire to daughter in inbreeding. Example of outbreeding, that is mating of animals of strains or species. Increase in homozygosity in inbreeding, whereas outbreeding increases heterozygosity. Inbreeding, it increases uniformity in progeny. Outbreeding decreases uniformity in progeny. Inbreeding reduces the variation, whereas outbreeding increases the variation. Okay? Inbreeding increases the breeding value, whereas outbreeding decreases the breeding value. Inbreeding decreases the reproductive efficiency, whereas the outbreeding increases the reproductive efficiency. Inbreeding decreases growth rate in progeny, whereas outbreeding increases the growth rate in progeny. Inbreeding decreases the production, whereas the outbreeding increases the production. Inbreeding decreases figure, whereas outbreeding increases figure. Inbreeding decreases disease resistance, whereas the outbreeding increases disease resistance. Inbreeding leads to inbreeding depression, whereas outbreeding leads to heterosis or hybrid figure. In difference between close breeding and line breeding. Close breeding means mating of closely related animals. Example are full brother and full sister. Line breeding means mating of distantly related animals. Examples half brother to half sister. Close breeding is intense form of breeding. Line breeding is mild form of inbreeding. In close breeding method, animals are selected for breeding on individuals' merit. In line breeding, animals are selected for breeding on pedigree method. Close breeding increases homozygosity quickly, whereas line breeding increases or reaches homozygosity slowly. Close breeding having both desirable and undesirable character develop rapidly. In line breeding, both desirable and undesirable characters are developing slowly. Close breeding it is most risky, while line breeding is less risky. Other methods of animal breeding, that is artificial insemination. The semen of the desirable male is injected into the female reproductive tract, called as artificial insemination. Multiple ovulation and improved transfer technology. The cow is administered with the follicular stimulating hormone to produce 6 to 8 ovules, that is multiple ovulation. This female is crossed with the desirable with the male and female. Male and the zygote of the 8 to 32 cell stages are placed in the surrogate mother. Then cloning. Cloning refers to the production of genes or cells or organisms with the identical genetic constitution. Clone is a population of cells or individuals which are genetically identical. Then cloning may be different types having gene cloning or cell cloning. First one is gene cloning. Multiplication of species gene into several copies by recombinant DNA technology or polymerase chain reaction, that is PCR, called as gene cloning. Whereas cell cloning, production of specific cell types called as cell cloning. Example, production of hybridoma cells that produces monoclonal antibodies. Organism cloning, production of individual plant or animals with identical genetic constitution called as organism cloning. In higher organism, cloning depends on totipotency, that is ability of single cell to divide and regenerate into complete individuals. Plant cells usually possess totipotency ability, but in animals only zygote possesses this ability. J.Y.Mote in 1997 from United Kingdom produces a clone of adult lamb and named it as Dolly. The procedure involved the following: isolation of udder cell, that is vegetative or somatic cell, isolation of egg cell from a female sheep. Then the nucleus is removed from the egg, called as denucleation. Denucleated egg is fused with the udder cell by the electrical stimulus to get a fusion product. Similar to fertilized zygote, the fused cell with the impulse of an electric current is forced to divide it under in vitro conditions. After a few divisions, the blastocyte is formed. This blastocyte is implanted in the uterus of the foster or surrogate mother. The lamb produced by surrogate mother is genetically and phenotypically identical to sheep from which the udder cell were used for cloning. These are the questions on this topic. Thank you.